So first I need to thank Peter for his 10 minutes of his talk. And the organizers and disorganizers for inviting an interloper into their midst. And I will try and convince you in the next 14 minutes and 59 seconds that I really do belong here. And basically sort of go through some of the history, and in fact, a lot of stuff which is not my work, but at the end, assuming I can get thrown off the stage, I will talk to you a little bit about some of our most recent stuff. But I really wanted to start out with saying it's really an honor to be here at a Phage meeting, because a lot of times people talk about standing on the shoulders of giants. Well, what we do in the archaeal virus community is try not to fall off. So basically, start out, I realize it's now 41 years that we've had archaeal viruses. They were, of course, first described as bacteriophage, and they were found during an investigation of flagella. So I always think this is kind of a neat way that you can find viruses when you're looking for flagella in the first place. Well, why weren't they called viruses of archaea? Well, it's because archaea hadn't even been discovered yet, or at least not recognized as such. That was by Carl Woese and George Fox in 1977. And so, again, not surprising they were called bacteriophage. But if then you continued to look at these viruses that were infecting extreme halophiles, what you found is most of those viruses actually kind of looked like bacteriophage that people had already seen before. And this was over the next, probably about 20, almost as many as 30 years. Almost all of these have pretty regular kind of head and tail bacteriophage-like structures. We'll ignore this little outlier over here for the time being. So it sort of made sense. And if you looked at the genomes, they didn't look like anything else. But Forrest already told us that's what phage genomes look like. Anyway, you find something you don't know what it looks like, you say it's a phage genome, right? So that was sort of where things started here. And probably the best work that was done on these viruses was done on the halobacterial phage, and it was called halobacteria for a really long time. So, and in fact, it's still called halobacteria, even though it's an archaean, done by Wolfram Zillig's lab. And I challenge you up here to tell me which one is P2 and which one is Phage, because they really are pretty darn identical in terms of looking at each other. But one of the things that Wolfram Zillig really did an absolutely wonderful job of was really buying into the whole archae bandwagon really, really early on. In fact, there was a meeting in Martinslied, just outside of Munich, which is where I did my postdoc with Wolfram Zillig, in 1981, where basically it was the first ever meeting where people were talking about archaebacteria and really trying to get a handle on how to work with these things. And to back up a little bit, turns out that Wolfram Zillig was interested in this not so much from the virus point of view. I think he just wanted to run around and poke around in hot springs. So this is really the pioneer, unfortunately passed away about 10 years ago now, Wolfram Zillig, now sampling in Yellowstone. And he had started out working on these viruses because he was really interested in setting up genetic systems. So I have to thank Zalio about saying that bacterial genetics and phage genetics were really pretty interconnected with each other. I think that was really the point of issue that Wolfram Zillig was coming from. He was trying to set up genetic systems in archaea, which at that point really didn't exist at all. And he'd in fact been working not only on bacteriophage T4, Phi-X174, looking at mostly transcription, but some replication as well. Many people don't know. I'll tell you a little quick story about this. He was one of the very first people to use phenol to extract nucleic acid from viruses. And one of his very first publications was showing that it actually was a requirement for nucleic acid in tobacco mosaic virus in order to actually get activity to come back. And then just a quick background. I wanted to have that wonderful first talk today. Put together your scientific background here. So Wolfram Zillig was my post-author advisor. Did his PhD with Wolfgang Butenandt, whose PhD advisor was no one other than Bunsen of the Bunsen burner. So I'm scientifically related to what all of at least the undergraduates here, maybe they don't use them anymore because they're not safe. But speaking of not safe, this is not a terribly safe environment either. But he was looking for these viruses to try and set up genetic systems with. And at first, he was expecting, OK, we'll find some nice, phage-like systems. And then, lo and behold, these lemons came out. This is a publication in 1984. Andrea Martin, and in fact, they originally found a plasmid, then later found a virus-like particle that was associated with it. These amazing lemon-shaped particles with a short tail at one end. You can tell a little bit by the scale here. Here's the sulfolobus. This is actually a pretty typical inhabitant of the environments that we just looked at on the last slide. These are the high-temperature, low pH environments. The optimal growth of this particular organism is 80 degrees Celsius and pH of about 3. So pretty extreme. We wonder if maybe some of these structures have something to do with it. But then he said, OK, well, this is one virus. We actually need a whole bunch more. So let's go and look some more. And this is really now going over probably, again, about 20 years of work, both from the Zillig lab and really continued mostly by David Prangishvili, with a whole zoo, and it's sort of a good introduction to what we'll be seeing tomorrow night, of various different virus morphotypes. And this is the most boring of them. These are the rod-shaped viruses. They're actually really quite fascinating because it turns out that they have a single-stranded DNA that's completely complementary to itself, or you can think of this as a linear DNA with covalently closed ends. But there's only 4 bases at each end before you start back on the opposite strand. How these things replicate, I've got some ideas, but I think is really pretty up in the air right now. Turns out that these viruses are actually pretty closely related to the filamentous viruses, also known as the Lipotrix viruses, that I was working on when I was Wolfram Zillig's lab, but also David Prangishvili did nice work with this. These guys are not only about twice as long as the cells that they infect, but they also have these really amazing nano-sized claws at the end here that seem to bind to pili, probably really important for the infection process. So these two, it turns out, have some sequence similarities between the two of them, the rudiviruses and the Lipotrix viruses. But probably my most famous is the one that was made for the birthday of Phage. These are the Acidionus bottle-shaped viruses, again found in these high-temperature, low-pH environments. Acidionus is very closely related to sulfolobus. These literally do have like a champagne bottle shape, but I think most importantly, at least for this birthday party, is they have all these birthday candles, about 20 of these little small filaments at the end of each of these particles. So really amazing diversity in terms of the morphology of these particular structures, but also in terms of what happens to these particular structures when they're outside the cell. Again, this is now David Prangishvili's work after he left Zillig's lab. He found these so-called bicauda which is when they're produced from the sulfolobus, which you can see here in the middle. You can also immediately tell these are lemon-shaped particles, but they're rather a lot bigger than those particles I showed you just a second ago. But when they're produced, they look like nice little lemons. But if you let them sit outside the cell for a couple of hours at 70 degrees, they grow these tails at either end. This is just a morphological change. The volume doesn't change at all. You can see this in the cryo over here on this side. But it turns out that it's only this one with the tails at the end is infectious. The ones here are not infectious. Quite how that process works, again, is a really fascinating question that we don't completely understand. Last one that I want to talk about, again from David's lab, is this really amazing virus which Tomohiro Mochizuki found, which is not only having this really amazing virion morphology, which is this open helix, but it's a single-stranded DNA virus that's over 22,000 bases in length. Now, how it's able to stabilize this under these conditions is, again, a huge open mystery. Well, I figured at this point, these guys are having way too much fun. I need to do this myself. So this is a picture of me in Yellowstone when I just started my postdoctoral work with Mark Young, second postdoc there, out searching in one of these really typical sulfolobus environments. This particular pool is 85 degrees Celsius and a pH of 3.5. Not terribly surprisingly, just like Wolfram Zillig and David Prangishvili, we find all kinds of really fascinating new virus structures when we're in there. This one's probably pretty similar to that bicauda virus here. Each of these individual particles with these tails. We didn't follow up on those. This looks more like a head and tail, but in fact with a lemon on the top. But what most important was this one right here. the STIV. And this one, again, little side story here. I was looking for lemon-shaped particles, and I was down at the electron microscope staring into it, and all of a sudden this icosahedral thing jumps off of the phosphor screen. I'm like, wow, I'm probably the first person in the world to ever have seen this thing. It was really pretty amazing. Now, why we're so excited about an icosahedral particle is, unfortunately, I think Jack had to leave, but his lab got this and did some absolutely wonderful reconstructions. This is the sulfolobus turritid icosahedral virus, so it was Liang Tang, we saw a picture of a little bit earlier on in Jack's lab. And they solved the structure. He actually mentioned T equals 31 in passing in his talk. This is that T equals 31 virus particle, icosahedral symmetry. We have no idea how all of those hexagons and pentagons get put together. Fascinating question. We have no clue. But what they noticed, probably even more important, at least from my point of view, is it turns out that the major capsid protein of not just this virus, but it turns out viruses from bacteria and viruses from eukarya have practically identical structures, even though the sequence similarity is undetectable. And that they showed first here by looking at these cryo reconstructions, but also in looking at the individual high-resolution X-ray crystal structures. And I think what that means is, of course, the last common ancestor of all life was infected by this, something like the virus that I detected. This is just that data here where we can see this is now the structure of the major capsid protein from our archaeal virus overlaid with the major capsid proteins of bacteriophage PRD1 and this case I think it was PBCV1. Another thing I wanted to mention here is that this virus, also very similar to PRD1, has a lipid layer underneath it, which we think may be important for the stability under these really extreme conditions. That was the first surprise that we had from STIV. But the second one, and this is work that was done after I'd been in Mark's lab and I started my own lab at Portland State, they continued and they were looking at the infection process and even more so the virus production process that happens with infection of, on the left-hand side here, the STIV. And what Mark's lab noticed, particularly Sue Brumfield, who's a fabulous electron microscopist there, is that there were these really amazing seven-fold symmetric structures that formed on the outside of infected cells and then burst when the virus came out. Even more bizarre is that basically at exactly the same time, David Prangišvili's lab in France was studying this rod-shaped virus, the boring one that I showed you right at the beginning, and it turns out these guys made identical structures to here, even though morphologies are completely different, the genomes are completely different, with the exception of one and only one gene. And it turns out that that gene, an expression of that one and only gene, actually will give you these really amazing structures here. Recently, David Prangišvili's lab has also come up with some really beautiful tomography. Each of these red bars here represents one of those rod-shaped viruses, and all the yellow ones here are these pyramids which have opened up and then released the virus particles, which have then come out of them. So at this point, it's like, wow, I discovered this virus and they find these really cool things about it. What's going on with the virus that I decided to continue to work with in my lab? Well, those are the SSV viruses, these lemon-shaped ones that I showed you right at the beginning, first found in 1984. I was really interested in them because, they're totally gorgeous, and this is my first ever electron micrograph, so I'm really proud of it. These are the individual particles right here, short tails at one structure, at one end of them, excuse me. And this is where also some of the similarities to some of the bacteriophage comes in. Turns out that production of these virus particles is inducible by UV irradiation. There's no obvious repressor protein. We've actually looked really hard to try and find one of these. So the process, again, is not terribly clear. Integrate specifically, not unlike lambda, although some really interesting differences that I'm more than happy to talk about a little bit later on. Probably the most surprising was that when you look at the genome that's packaged inside these particles, it's positively supercoiled. In fact, one of the very first examples of positively supercoiled DNA to be found in a naturally occurring state. But it turns out this is probably a great adaptation to surviving under these extremely high temperatures, because when you positively supercoil, you overwind your DNA and hold things together really well. So we're interested in all kinds of things. We do a bunch of genetics. I part of my undergraduate research courses we used to do some of the genetics. But I just wanted to talk really briefly about our latest work, which was published on the cover. I'm sure all of you actually look at covers of journals these days, right? And the cover of Virology, you know, on the 1st of January issue, is our structure right here. This is now a cryo-electron reconstruction done in collaboration with Mark Murray's lab at the UT Medical Branch in Galveston, Texas. A couple of important things to see here. One is that our resolution is nowhere near as nice as what Jack showed you right after lunch. This is only at about a 35 angstrom resolution, but a couple of things you can see. One, there's no obvious tightly packed DNA, which is present on the inside here. So the first thing. The second thing, it turns out that this does have a bit of a hexagonal structure to it, and we think what this structure actually looks like, and this is pure speculation right now, but it's a model that fits, is was mentioned earlier, I think by Alan, that to put together a closed surface, you have to have 12 pentagons. But they don't have to be arranged completely icosahedrally symmetrically. So we think what's happening here is we've got these two so-called fullerene cones that fuse in a manner, in a very broad sense, not dissimilar to what happens with HIV. Finish off, again, phage and archaeal viruses are really similar. This is a case of an SSV with a satellite virus. Here are P2 and P4. The other set of viruses you find in archaea are the methanogen viruses. Strangely enough, mostly look like what you see in terms of the halophile viruses, with this one down here that I think is actually kind of like STIV. Recently, Dennis Bamford's group has done a really nice survey of looking at halophile viruses. Again, most of them look like pretty classic head and tail phage, but you have a few that look here like the STIV-like structure. I can't end here without mentioning David Prangashvili's work. Again, most of the stuff I actually talked about today, particularly the zoo stuff, was all done by him, either in Wolfram Zillig's lab, Carl Stetter's lab, or in his own lab at the Institut Pasteur. Wolfram Zillig, as I mentioned, passed away about 10 years ago now, but really is an inspiration to me and I think a lot of other archaeal biologists. This is Carl Stetter, for those of you who don't know him. Finish off again, I think I've mentioned that Wolfram Zillig was really my inspiration for getting into this. Hans-Peter Arnold was a graduate student in the lab together with David Prangashvili as a postdoc when we were both there. Ingelor Holtz, Wolfram Zillig's partner and major partner in crime in terms of working with all of these things. And Monica Herring has been working in David Prangashvili's lab and discovered a lot of these brand new viruses. I did my second postdoc in Mark Young's lab together with George Rice and Blake Wiedenheft, Montana State University. Talked about some of Jack and Dennis's work, and then Mark Murray did all of our structures. These are people who've been funding work in my lab, and with that, I'll take questions. Lynn.
Viruses.
Yeah.
Is the growth a mechanical or physiological metabolic?
So the question is basically, what is that change which is happening there? It seems to be a purely mechanical process that takes place. So there must be some kind of metastable state that you have in the sort of compacted football form that then gets stretched out. And they're looking at in fact, we know the proteins that are doing that, or should they know the proteins, and so they're looking at how these guys are polymerizing with each other. But it does seem to be purely mechanical. There's no metabolism that seems to be taking place at all.
Am I correct that you very seldom see these viruses actually free in nature, that they spend most of their time as prophages?
So yeah, that's. Do you ever see them just hanging out by themselves? In terms of these hot springs, it turns out that the virus numbers there, we've actually tried to take a page out of Jed's book and do some staining here. It turns out that the numbers are incredibly low in some of these systems. And in fact, Forrest and Maya know this as well. But as soon as you look at some of the hypersaline environments where you've got a lot of these viruses, you see huge numbers there as well. So it may just be the high temperature, low pH environment where they're not very happy. But we're very interested in getting higher resolution structures to figure out what does make them happy. It turns out that those particles, particularly the lemon-shaped particles, we can leave them at these high temperatures for weeks, and they're actually pretty happy. The low pH seems to be more of a problem.
So Kim, with the pyramids, did those burst at the same time, or did they? I mean, when
So what happens with the pyramids? Now, okay, I'm getting out on a limb here because this is not my work. It seems that they open more or less simultaneously. You get pretty similar opening. What the regulation of that process is, I think, is pretty open. But as I mentioned before, you can take that one protein, you can express it in E. coli, you can express it in Saccharomyces. It makes pyramids, but they don't open. So there's something else which is going on there to get that final process to take place.
Thanks, Ken.
Thank you.